In this case, the amnion, and on the other side, the yolk sac. This thing now starts to grow in size, you can see. Here again is this embryonic disc consisting of 2 cell layers. It's flattened, yolk sac and amnion. And then it goes through a very important stage, which is called gastrulation. It's here that you get a development of what are called the 3 primary germ layers: ectoderm, mesoderm, and endoderm. Up to this point, the cells are basically the same. But at this point now, they differentiate into different cell types, which will give rise to all the tissues and organs in the body. Those so-called 3 primary germ layers. So I'd like you to know ectoderm, mesoderm, and endoderm. And again, the process is called gastrulation. So again, this inner cell mass flattens and becomes an embryonic disc. And it is bilayered. It's got 2 cell layers. And that's what you see right here and here. Okay? What's then going to happen is gastrulation. And it's here that the cells begin to differentiate. Now, what we have here, again, is what we saw before. The bilayered embryonic disc. Below that, the yolk sac. On the other side, the amnion. And right in here, the 2 layers, that is the embryo, the bilayered embryonic disc. When you look at this, you see here's the amnion, here's the yolk sac. If you make a cut through that, and then look down on the top, what you're doing is seeing this. This would be the head end, what becomes the head end of the embryo. This is the tail end. There's a little slit here, which is called the primitive streak, close to the posterior end. What's going to happen is, in gastrulation, cells are going to migrate across the surface of the embryo on the amnion side, pass through the primitive streak, and spread out below. This is gastrulation. By the way, it's on the amnion side. And then they spread out below. The first cells to do so give rise to the endoderm. The next cells and migrate into the primitive streak and sprout below, give rise to mesoderm. the cells remaining on the surface become the ectoderm. You can see them here. Here again, we're looking at this. We're looking at it from the top. So like you're cutting through, looking down on the top of the embryo. This would be the embryo side, and below would be the endoderm side. Now you're taking a slice through this, and you're seeing this, okay? And here is the primitive streak, just the amnion side of the embryonic disc, the embryo. The first cells that migrate across the amnion side of the embryonic disc and pass through the primitive streak and pass out, spread out below, endoderm, shown in yellow. The next cells that come in move across the surface of the embryonic disc, amnion side, and pass through the primitive streak and spread out below, mesoderm, and they're shown in red. The cells remain on the surface then become ectoderm. So now you got your three primary germ layers. Ectoderm on the surface, mesoderm in the middle, and endoderm below. They're called the primary germ layers. These will give rise to all the tissues and organs in the body. And I've listed a process called Orogenesis. And I've listed some of the tissues and organs that develop from the ectoderm, the mesoderm, the endoderm, which I'd like you to know. Generally speaking, the tissues on the surface of the body, or structure on the surface of the body, or near the surface, are of ectodermal origin. This would be the epidermis, the skin, our sensory organs associated with the epidermis, sensory receptors, our nervous system, brain and spinal cord, and our nerves, all develop from ectoderm. And that's there in your notes, as you can see. Epidermis, the skin, nervous system, sense organs, and then things of epidermal origin that are associated with the epidermis, like the fingernails, the toenails, the hair, and sweat glands and oil glands, for example. Things that basically in the middle of the body develop from mesoderm. You can see this would include our bones and cartilage of the skeletal system. It would include our muscles. It would also include the dermis, the skin, our reproductive organs, the organs of the excretory system, and most of our connective tissue. And up at the top it says notochord. Notochord is actually a supporting rod that develops within the embryo. In fact, it's one of the characteristics of our phylum, to which we belong, the phylum Chordata, which is a supporting rod called a notochord, a dorsal hollow nerve system. And in our brain, of course, we got cavities called ventricles. And we got a cavity that runs down much the length of the spinal cord called the central canal. It's a hollow cord, okay? And then gill slits. And actually, we have gill slits early in the embryo. Normally they close. In fish, they often remain, but not usually in humans. That's actually a characteristic. But the notochord is a supporting rod that precedes the development of the vertebral cord, or the vertebral column, which we have in vertebrates. Okay? Any questions on this?
The cells differentiating into the endoderm, mesoderm, that's called gastrulation?
Yes, the initial differentiation of the germ layers is called gastrulation. Which I described it on the board, with the migration of cells across the surface of the embryonic disc into the primitive streak and split out below. That's gastrulation.
Okay, and then what is the organogenesis?
Organogenesis is formation of the tissues and organs from these germ layers, which occurs later.
The gastrulation is just the differentiating of the cells, and then the...
Gastrulation, we get differentiation of the cells, that's right. From the germ layers, and of course the germ layers will form these different organs and tissues
and then
later.
actually turning into epidermis and all that is organogenesis.
Mhm. formation of organs and tissues, okay, later on. Like I said, up to gastrulation, the cells are basically the same. There's no differentiation. As you also probably know, all our normal body cells have all the genetic material in their nucleus. It's just that genes get shut off and turned on, depending on what the cell is doing. And when those genes don't turn off, then you have problems. And you end up with abnormal cells like cancer cells, especially those associated with cell division. Because one characteristic of all cancer cells is they continue to divide, unlike normal body cells. Okay, again here, this is continuing, this is figure 2810. What we saw before, looking down on the embryo, from the amnion side of that embryonic disc, there's a primitive streak, gastrulation involves migration of cells for example. What eventually happens is, as you can see now, we got ectoderm, we got mesoderm, and endoderm here. This is yolk sac and that's amnion. But now we've got the three germ layers and things are going to start to develop in it. This is the notochord. The notochord is a supporting rod found in all chordae embryos, including humans. Okay? Eventually, in vertebrate animals like ourselves, that's going to be replaced by the vertebral column. You can see this thing continues to develop. And what happens here on the surface, the ectoderm, which is in blue here, is going to form a groove. That's called a neural groove. Eventually it's going to fold over and make a neural tube, which can develop into the brain and spinal cord. And we have a dorsal hollow nerve cord. That's one of our characteristics. And again, it's hollow because in our brain, remember, we have the ventricles, continuous with the central canal of the spinal cord. This, of course, is going to give rise to various connective tissues, give rise to our muscles, to our bones, cartilage, and so on. And this endoderm is going to give rise to GI tract, okay, our gut, also organs of the digestive tract like the liver, the pancreas, and so on. I forgot to mention these, but you can see them there listed. Digestive system, respiratory system, liver, pancreas, and pharynx, which of course is your throat. Common to both the digestive and respiratory system. You can see this thing continues to develop. Now you've got a neural tube, and that is going to become the brain and spinal cord, dorsal hollow nerve cord. There again is the notochord, but eventually it's going to be replaced by the bone and the vertebral column, invertebrate animals, and then more of the same. You can see. Now the endoderm, you can see, is forming what? The gut, which is going to be the GI tract. It is going to give rise to muscle and bone and so on. Okay. I'm not going to quite know all this, but I'm just showing you basically the steps that go through. But again, it all comes from what? The three primary germ layers. So I want you to know up through those layers. The formation of germ layers, which occurs in gastrulation. Very important phase in the embryo. And then I'd like you to know what develops. That's in your notes from these three primary germ layers. Ectoderm, mesoderm, and endoderm. Okay. Any questions on this? Here again, we're looking at now from a different point of view, side. Again, you can see here's the amnion, here's the yolk sac, here's the three germ layers, ectoderm, mesoderm, endoderm. Now the embryo you're looking at from the side, the tail end and the head end. See, it folds over. forms the embryo. You can see, look at it from the side again. Here again, you can see from the side, cut longitudinal section. This would be the dorsal hollow nerve cord, brain and spinal cord. This is the supporting rod, the notochord, which is from the mesoderm. This coming through the yolk sac is going to be what? Your GI tract forming. And also organs of the digestive system, like the liver, pancreas, and so on. Okay? Eventually, of course, now, I want you to know those extrinsic membranes. There are 4 of them. The chorion, and the chorion develops from the trophoblast. The chorion is also going to form part of the placenta, which we can see right here. And you're going to have folds of the chorion, basically,